Hi, Assalamualaikum. My name is Nurfana Shamimi binti Muhammad Shukri with the student ID 2024697978 from program code of AS239 and currently taking the course code of FSC 631. So this is my title of the research, which is the assessment and modeling of airborne biological contaminants, bioaerosol, and their association with indoor air quality and health indicators in educational building. So now let's look into the background and also the research problem of the study. So, for introduction, according to Nabipu et al. 2025, exposure to bacteria and fungi through inhalation may trigger various immune response, including allergic reactions, infection, and chronic inflammation. Individuals that are more susceptible, such as children, older adults, and those with existing underlying respiratory or immune conditions, are at higher risk. And according to Zhou et al. 2025, studies shows that the concentration of microorganisms in indoor air influenced by a variety of factors such as season, temperature, relative humidity, and at the air exchange rate. So for a problem statement, we will look into the limited understanding of biological contaminants and their health effects, and also lack of predictive regression equation for biological contaminants. And let's look into the research gap, which we will like to address the limited studies on educational building, limited studies assessing biological contaminants, and also lack of predictive regression equations for biological concentration based on relative humidity, temperature, and also carbon dioxide. The objective of this study, the first one is to identify the concentration of bacteria and fungi in educational buildings, specifically in Faculty of Applied Science UITM Shah Alam. The second one is to evaluate the health risk assessment of students and staff at educational building. And the third one is to evaluate the effects of concentration of bacteria and fungi on occupants' health symptoms and subsequently develop a predictive regression equation for bacteria and fungi concentration. So next, let's look into the research question. The first one, what are the concentration of airborne biological contaminants such as bacteria and fungi present in educational building? The second one we look into, does the concentration of bacteria and fungi associated with IAQ parameters in educational building? And the last one is, can a predictive regression equation be developed to predict the concentration of bacteria and fungi in educational building? For methodology of this study, the study will be conducted at the Faculty of Applied Science, UITM Shah Alam, specifically in the classroom, auditorium, library, laboratories, and also staff offices. Next, we'll move on into preliminary workroom, in which this will help to gain overview of the sampling location, identify potential sampling points, and sources of biological contaminants. Next, for the sampling point, in determining the sampling point, we will be decide the sampling point based on the guidelines from ICOP 2010. And next, preparing agar plates, in which we will be using two types of agar plates in this study, which is the TSE, tryptic soy agar for bacteria, and also MEA, the mild extract agar for fungi. And then the sampling procedure will be conducted, which it begins with calibration that will be made by the laboratories, the sampling, and also the incubation of the bacteria and fungi. And then for the distribution of questionnaire, which is will be will be help in health risk assessment, which will be given to the population to help identify health risk symptom and identify the occurrence of sick building syndrome. Next, for the data analysis, in which the concentration of bacteria and fungi will be identified and calculated and expressed as colony per cubic meter or CFU per meter cube and compare with the acceptable limit in ICOP 2010. And lastly, form a simple regression equation which will be formed to predict the concentration of biological contaminant based on independent variables such as relative humidity, temperature, and also carbon dioxide. So, what are the expected outcome of this study? The first point, we will review the concentration of bacteria and fungi in Faculty of Applied Science. And we look into the association between bacteria and fungal concentration and health symptom among students and staff. And then we will look into the association between other indoor air quality parameter with concentration of bacteria and fungi. And lastly, to develop regression equation to predict the concentration of bacteria and fungi from independent variable. And what are the significance of this study? The significance of this study, the first one is to understand contributing factors growth of bacteria and fungi in educational building. The second one to improve a healthy educational environment. And the last one is to provide tools predicting biological contaminants in educational building.